So today we're going to be talking about how glucagon affects the metabolism of amino acids in the body. So at any given time, there's a certain amount of free amino acids flowing in the bloodstream, and this is due to the breakdown of proteins that have been consumed and digested in the digestive tract, and through the breakdown of proteins in the body when they're no longer needed. So these free amino acids have to go somewhere, so there are multiple options. They can either be used for growth or the creation of new proteins as needed, or transformed through the process of gluconeogenesis and synthesis of other amino acids. Today we're going to be focusing on how they're excreted particularly through the urea cycle, which takes ammonia that results from the breakdown of amino acids and converts them to urea, which is far less toxic. And this ultimately, the urea is excreted through the kidneys and through the bladder. And this process mainly takes place within the liver. So you may be wondering right now how glucagon is involved in all of this. So glucagon is a peptide hormone that is released by the alpha cells in the pancreas. And traditionally, it's always been paired with insulin because it's best known for increasing blood glucose through glycogen breakdown and gluconeogenesis. However, the results of recent studies have suggested that glucagon may have a greater involvement in the breakdown of amino acids. So it's been shown that when the glucagon receptors have been knocked out, the alpha cells in the pancreas have further proliferated, and it's believed that this was directly due to the increased presence of amino acids. In this particular study, the researchers hypothesized that a feedback loop exists between glucagon and amino acids, in which glucagon increases the breakdown of amino acids, while amino acids, on the other hand, stimulate the secretion of glucagon from the pancreas. So this study was performed using mice that were wild type and mice that had their glucagon receptors knocked out genetically. So while normally they were given food ad libitum, these mice were fasted for 4 hours before performing tests. During that time, they were not provided with any food, but had free access to water. Afterwards, they had blood drawn, and their pancreases and livers were harvested, and then later their blood was drained and then sent for tests. So in the next part of the experiment, all the mice were wild type and were still given the fasted for 4 hours before their organs and their blood was harvested, but they were given very different treatments beforehand. The first group was given no treatments. They were simply injected with PBS. In the second group, they were given an amino acid injection known as Vamin, which is just a commercial mix of amino acids, right before they were given before they were fasted for the 4 hours. In the third group, the mice were given a glucagon receptor antagonist before they were fasted. And then in the fourth group, they were given both the amino acid injection and the glucagon receptor antagonist. Analysis of the blood from these mice revealed that compared to wild-type mice, the mice that had the glucagon receptor knocked out had lower glucose levels. This isn't surprising because of glucagon's well-known role in raising blood glucose. However, it was also found that there were higher levels of amino acids in the blood, as well as higher levels of glucagon itself. This increase in glucagon can be explained by the findings of the pancreases of these mice. The islets, the groups of cells, the groups of alpha cells in these particular mice were about 6 times as large as they were in the wild-type mice, and further analysis showed that there was more mature glucagon. In the wild-type mice, there was more pro-glucagon, which is the precursor of the mature glucagon, which gets released into the bloodstream. So this indicates that there was far more higher production of glucagon in the knockout mice than in the wild-type mice. So when additional amino acids were given to wild-type mice, they were found to have higher glucagon levels as well. However, the analysis of their pancreases did not show the hypertrophy that was found in the knockout mice. In addition, when the glucagon receptor antagonist was added, the amino acid levels in the blood also increased, although there was still much less hypertrophy than was seen in the knockout mice or in the mice that were stimulated with additional amino acids. However, the conclusion that amino acids stimulate the release of glucagon itself comes from the addition of vamin, or the mix of amino acids, that were given to wild-type mice, because there was no other explanation for the increase in glucagon levels. Now, when the antagonist was added, it would be fair to assume that the glucagon levels would increase due to the lack of receptiveness of the liver and other organs. So based on that, it would be alone, it could be fair to assume that the increased glucagon was directly responsible for the increase in amino acids. This would also be a fair conclusion of the results from the knockout experiment. So this figure is showing the results of the blood test from the experiments where either the mice got no treatment, or they got amino acids, or the glucagon receptor antagonist, or a combination of those two. Naturally, when more amino acids were added, the amino acid levels increased, although they did decrease over time. However, when both the antagonist and the amino acids were given together, the levels of amino acids increased substantially more, which is showing a synergistic effect, which is consistent with the feedback loop that's been presented. So the figure on the right is showing the glucagon levels over that time as well, with the same treatments as previously stated. So when the glucagon receptor antagonist was given, the levels of glucagon initially increased, but then went below the control levels around 12 minutes after administration. When amino acids were given, the glucagon levels once again peaked, but then eventually decreased over time. When both the antagonist and the additional amino acids were given, the glucagon level initially spiked around minute 2, but then decreased to close to normal around 20 minutes. These findings once again are consistent with the model that glucagon stimulates the breakdown of amino acids, and that when amino acids get increase, those stimulate the increase in glucagon secretion. So the first discussion question is, what would the results be if a glucagon agonist had been used rather than an antagonist? By results, that can include the changes in glucagon levels, or amino acid levels, or even the effects on the pancreas itself. The second question is, what metabolic changes may have affected the results that were obtained with the knockout mice? Here are my references, and thank you for listening.